Yeah, well, what I'd like to talk to you about today is our simulations that we do for cellular processes. And you'll see examples where we go from single cells to colonies. We haven't quite got the colonies onto blue water yet, but we're on our way. And to do these kinds of problems, you know, I have to make really use of the fact that I have appointments in like several different departments, because you need physicists, you need computer scientists, you need biologists to help you solve these problems. And much of the work that we do on the cell simulations has been motivated by our NSF Center for the Physics of Living Cells and trying to keep ahead, or at least even, with our experimental colleagues. So when we're looking at biological modeling, I think it's really clear the problems exist at all sorts of different levels. And so the interactions, they span magnitudes, several orders of magnitudes in space and time. And you've heard a lot at these meetings about molecular dynamics simulations, the potential-based, all atoms, enormous amount of work goes in developing those potentials. You can try, the time steps are like femtoseconds. You're looking sort of in this regime. If you want to go, let's say, down to times that are relevant for cell procedures or cell processes, we need hours. So you can try core screening, or like with Brownian dynamics. But what I'm going to tell you about today is we use a probabilistic approach to solving these problems. And it's called this reaction-diffusion master equation. So we're really focusing on calculating the probability of the cell to be in a particular state. And again, I point out the length scales are terribly different. Even if you're doing like subcellular systems, you're in the hundreds of nanometer range. You get to the cell, you're in microns, several microns, and if you move it up to large populations, you're getting up into the centimeter range. So again, this shows you sort of where each of these methods extend and how they overlap. And of course, we'd like to, if possible, make contact with one another and use a lot of the work that has come from molecular dynamics, where possible, to inform us at this sort of level. All right, so how are we treating the evolution of the cellular state? As I said, we're calculating its probability. This is this P here, and X stands for the state of the cell. That can mean so many particles at a certain time, and this is a multidimensional vector, so it's RNA, DNA, proteins, perhaps particular sugars that you're interested in. And if you think the cell is well stirred, then you don't have to worry about diffusion, and you just calculate these reactions. And we use a stochastic integrator for that. So those are like, the A's are these propensity expressions. So again, really high dimensional. If you take into effect that the cell is really a very crowded and noisy place, a lot of heterogeneity. In the bacteria, it doesn't look that bad, but if you go to a eukaryotic system, you start having at least 16 different compartments and boundaries that you have to cross in going from one to the other. So to that reaction operator, you have to add a diffusion term. And if you think about packing all this into all those macromolecules into a cell, it looks pretty dense. And when I first saw this, I go, oh, how does anything take place there? And then you take the slice and turn it on edge. So it is about 50% packed once you put in all the proteins, DNA, RNA. But there are these open spaces where things are diffusing and reacting. So this is a real problem, right? And what I'll show you are just a few quick results on problems that we've looked at. It's not running, so we'll make it run. One is on, well, what the heck. All right, this cell will divide if we wait long enough. And in doing that, it sets up an oscillation of chemicals. And here I plotted that this as a function of time. And here's the midpoint of the cell. And you can see there are fewer that are there. And that's what the point is to keep that midpoint region free so it can pinch itself closed. And down here, these movies should not run, but they are now looking at a problem of stochastic gene expression, because no one cell behaves the same. There are going to be different amounts of proteins in them, and that can lead to different fates. Now scale this whole thing up. These are all independent cells. If you now have a whole collection of cells and they're competing for resources, whether that be oxygen or food or metabolites, then there's going to be an overall collective response of this colony, and that'll lead to have the cells in the interior having a different metabolism. That means like they could be taking up and secreting different particles than the ones on the outside. For those of you who have never seen a metabolic map, this is a cartoon of the sort of core mechanism. So most of you maybe hopefully in school have learned about the glycolysis reaction, the TCA cycle, those type of things would go in there. And in the first version when we solved this problem, that's an awful lot of reactions to be considering. So we just had reaction diffusion on the outside that updated the steady state solutions on the inside of the cell, and we fed it back and forth. All right, so now just to quickly summarize everything I've been telling you, it's all bundled together in our software called Lattice Microbes. It is allows us to do a sampling of this reaction diffusion master equation to look at reactions within in vivo cells with a proper amount of molecular crowding. I'll tell you in a moment how we get that crowding, but this is how it looks. It's packed, and if you go over and use what they call proteomics data, you can see there's a wide distribution of sizes and percentages. Add in DNA and you're getting up to 45 to 50% of the cell being packed. Now this movie is supposed to run, so let's see what happens.
Seems to be loading.
Yeah, it's just very slow. Okay, well what we could do is, man, this hasn't happened before. Oh, good Lord. Huh. If you excuse me, the one last time that this helped was just to kill PowerPoint and start it up again.
We do that on Blue Waters too.
Yeah, I know. I get the report.
It doesn't work quite
See, yay! Success! All right. Yeah. All right, so this reboot went really fast and didn't cost a lot of money. So you see the clock ticking here? So that's what I'm saying. When you're doing these cell simulations, if I'm trying to look at stochastic gene expression, so you've really got to be able to simulate over several cell cycles to be able to do this. And these little blips that you're seeing are messengers being formed, and then those messengers are translated and give you proteins. This cell has really turned on, so is this one. These others don't have very many of those proteins on the membrane, so they're not funneling in the food, they don't switch on. This has been studied experimentally, single molecule work, so we were able to use that data to validate our approaches. Did we get the right time course? Did we get the right percentage of cells turning on for any particular concentration of sugars on the outside? So here's a little cartoon. Here's your DNA. That DNA, there's a repressor sitting on that gene that makes that protein there. If you start putting in sugars and these membranes also funnel in the sugar, it can help decrease the binding of the repressor to that gene. And when the gene is now on, it can make the messenger. The messenger then goes, protein gets translated, and so it gets really switched on. But not every cell is the same, as you see here. And that's one of the problems of biological biophysics. In fact, I had a colleague, we just came actually from the NSF in Washington, D.C., where we were talking with other physics frontier centers that like do cosmology or gravitational waves. And I said, you know, the difference between what we do in the Center for Physics of Living Cells is our systems fight back. You know, if we do something to them, they change. They never get the same response to them, whether you examine them experimentally. So you have to really do statistics. Look at cells individually. The average just will not do for you anymore. Now, back to our overview of our cell simulations. We really tried to bring in as much experimental data as possible. So, for example, about the cell structure, we bring in these tomograms, those cryo-electron tomograms. This is from one of my collaborators at the Max Planck. So you see, you get the size of the cell, you see these dots, those are the ribosomes, or some of the biggest crowders, and you also get a faint outline of the nucleoid region, where the DNA is sitting. This is very important data. From proteomics, that can give you that long list of other particles that are in there, and then the whole field of biochemistry has just generated enormous information on the reactions that go on in the cell. They've studied them individually, and even the rate constants are stored now in a database that actually sits in Germany, in Braunschweig. So you put in all this information, you set up your system, and then you send it off to either a GPU cluster, but if you really want to get things done quickly, you send it to Blue Waters, because it can only do, this is the only machine that will allow us to really put in all the information. And then you simulate, and then you send it back. So here I just put this up to remind people, we are really, really happy with the GPUs that are on Blue Waters. They're fast, they're efficient, we like them, we only wish there were more, right? Okay, so one of the problems that we have solved, and this is where we used our multi-GPU code, is to look at the response of a colony for a competition, say for foodstuff, how that affects the metabolism of the individual cells. So the way this model goes, you have the RDME model, it's doing diffusion and uptake of the extracellular sources, these things out here. It then controls the fluxes into the cell, but we can't really do a time study of these, but we look at, for that given uptake, what would be the steady state a solution to, say, fluxes through various parts of that network, and then are they secreting or taking up something like oxygen or acetate or glucose, and then we feed that information back into the reaction-diffusion equation. So now you'll see, hopefully, a short movie of this. Yeah, it's working. So here's at the edge of the colony, so you're funneling in oxygen and glucose. Oxygen is diffusing very quickly, glucose is coming in, but what happens is the cells that are closer into this region suddenly get deprived of oxygen, and they go into some metabolic state where they start secreting acetate, right? So now we've scaled this up from these few hundred cells to millions of cells, and now can look at this also experimentally to verify that there should be this region where you see acetate if you put an acetate marker in there, something that can report the system. All right, just to give you a feel for our multi-GPU code, and again, we're indebted to information that we've obtained also from NCSA on some of their previous machines to Blue Waters. There, we were lucky enough they had machines that had multiple GPUs on a node. And so now we can break apart any organism into these multiple GPUs. And again, I want you to see the cell finally divide. So these neighboring GPUs are sharing information. So the one in the center, or say 3, gets information from 4 and from 2. There's a sort of a shadowing region there. And I would like you to notice it's pretty good scaling. And the only thing that's changed between these sets of curves and these sets of curves is when we put a lattice over our system and do our simulations, we've changed the resolution of that lattice. So these were like 8 and these were 16. And when we did the colony that you saw, those were 20. And they were in this region. So this is what we've been busy for about a half a year now trying to implement this multi-GPU code in the MPI version onto Blue Waters. And this is our estimate of our best performance that we think we will be getting on it. We're not quite done. And the reason we're optimistic is because if you look at the way we've programmed it, this gives you sort of a time step in our multi-GPU code. So here's the guy in the middle. He's getting information from the left and from the right. And when you're using these single nodes but multi-GPU systems, you have these peer-to-peer transfers. But now if we go over here to Blue Waters, what we're using is that GPU direct, where we can exchange information from the GPUs that are on the multi-nodes, but we don't have to bother the host GPU. And so this is what we're trying. It's looking pretty good. We just didn't get everything done for today's lecture. But I will show you what we have achieved so far. This is a very important problem. It's one of the fundamental problems in a cell is ribosome assembly. As you know, the ribosome is where the proteins are made. There's the large subunit, and here's the small subunit, that's RNA both places. And these are about the 50 proteins that are around it. Of course, as theoreticians, it's all just spheres and lines. And so this wiggle thing is RNA, and these 50 proteins have to bind to it, or if we just look at this small one, 20 of them, you could have up to 2 to the 20th states. So what we do is we have to reduce this, and we have to reduce the number of states we have by using experimental data again. And this is a result of our reduction. This is a curve obtained from the dots or the experimental curves from in vitro assembly, and the dark lines are ours. And we can use several reductions of intermediates and reactions. And the idea is to start off with some initial distribution of ribosomes and see how it would get to a new state of the cell. And let me show you how the experimental data we use. There's a very well-known assembly map which tells you that the ribosomes assemble in sort of a direction, first at one end, and then the middle, and then the next, and then the other end. And there's certain proteins that have to be in place before the other ones can come in. That has been reported originally by Nomura, but we've also done experiments and calculations with colleagues to verify parts of this. So what it tells you, of all the states, we can knock some of them out, and this drastically reduces what we have to look at, say, in that beginning sector. Now, we use that information. We predicted in vitro what the intermediate should be, the most populated ones, compared them to the experiment, and it agrees extremely well, or I wouldn't show it here, even though the paper is not quite written yet. And what's interesting is we can resolve this end part of this assembly. This is cryo-EM work over here, so you can't see all the intermediates. We can see more. And this is our binding order. So we're starting with sort of the naked RNA here, and we're adding proteins according to our kinetic model. And if you see the gray lines, that's the full system with over 1,600 species and 7,000 reactions. And you'll see this, most of the flux goes through a very few number of intermediates, and that's what we'll use in trying to go to the complete in vivo model with all the other cellular networks. And we have tested these intermediates also on blue waters by using NAMD and trying to understand the simulation. So now I'll just end with our blue waters movie. This is the cell. You see the nucleus here. There's some genes on the DNA. We had original distribution of ribosomes to the end, a little unphysical, but we did it to demonstrate nicely the assembly. And now you're forming the ribosomal proteins. You have some equilibrium state. The clock is ticking. And at the end of this simulation, you'll notice now you'll have more ribosomes coming in where there's space at this end and along the side, and 220 ribosomes have been added to this. Right. What was really important being able to do this, because there's some uncertainty even in the literature about the rates that you should be using this. There are many reasons why to use blue waters. One is our software is inherently, it's a GPU based, and we can exploit the blue waters architecture. This is a large problem. It's going to need significant computational time. And these spatial models are larger, at least, than anything we've done. And I think it's, you know, I was looking at people who do explosions, maybe they get more reactions. But this is a large number of reactions to have to do, and so we're really trying to do everything we can to reduce the number, because the compute time is proportional to the number of reactions. If we can use a core system of reactions, we can drastically reduce our time. But what's also critical was using blue waters to do a parameter sensitivity analysis. So we took 560 nodes, we did 500 replicates, 25 different parameters each, and we generated these large terabyte of data that we then analyzed on the nodes using a Python interface to reduce it to 200 gigs so that we could transfer it to our local machines. So our end goal is really this was just translation I showed you, connect it to metabolism, connect it to regulation networks, and do a full model. And one of the other real bonuses of Blue Waters is that I/O hardware keeps pace with our simulations, which is really important. So here's where we want to be when we're done with this complete simulation, is combine all these components of the cell with the metabolism. So we want to do assembly, transcription, regulation, and use reduced systems for each of these networks. And that will allow us to see how the environment perturbs the system, and that perturbation then will be demonstrated in terms of growth, what gets secreted, or the motility of the cell. So a lot of what I showed you is contained in these references, which are also at the end of the abstract. I won't dwell on that. Instead, I'll just show you who are those people who have done it. So one of the lead computer science researchers helping us is Mike Halleck. He's bringing our multi-GPU code that we published in Parallel Computing to Blue Waters. These students are also helping with the transfer, and Kei Chen was a very fearless postdoc who really drove this one particular to do this whole network, and it really made us go to larger system, larger number of equations, and made us think how to change it. And of course, our visualization and getting our code onto these GPUs was helped enormously with John Stone and John Cole, and there are experts on systems biology and connecting it to the metabolic network. And Jonathan Lay was in charge of looking at these interesting intermediates in the ribosome assembly and studying them using NAMD on Blue Waters. So between us and between Lattice Microbes and the NAMD, we're eating up all of our Blue Waters allotments. And of course, we're really thankful not only to NCSA, but NSF for helping us with our center, but also getting us Blue Waters. And our early runs on using multiple GPUs were done at Keeneland, so we have to thank them too, and of course our exceed allocation. All right, thank you very much.